Okay, so what I want to do here, as briefly as possible, is try and look at some of the philosophical implications of endosymbiosis and symbiosis in general, but focusing on endosymbiosis and its implications for evolutionary theory. And I think an easy way to set this up is to look at these two historical books and compare their messages. So the top one is Jan Sepp, who's a historian of microbiology, of symbiosis and endosymbiosis in particular. And Jan Sepp gives a long history from the 19th century onwards of anti-Darwinian associations. So thinkers who thought endosymbiosis and symbioses in general have raised really serious concerns for neo-Darwinian thinking. Of course, obviously this has to come after Darwin, but people start thinking about symbiosis in relation to it. And Sepp himself, as I'll show later on, also begins to think that that these problems are almost insurmountable. And I want to contrast that with John Archibald's view. So John has this beautiful new book that many people here have already read, and if you haven't, you should. And the message John has there, which is a bit more implicit than Sepp's, is there are minimal theoretical implications for Darwinian thinking from endosymbiosis and symbioses, but they're similar to those of lateral gene transfer. We want to acknowledge lateral gene transfer, we want to include it as a phenomenon, but it doesn't perturb the theoretical apparatus. And I hope I've got that right, John. Just contradict me in the questions if not. So, just starting historically, and I've got no time to do this. If anybody wants sort of a more historical perspective, they can go to John's book or to Sepp's book. So we have one of the first evolutionary statements about symbiosis and endosymbiosis in particular from Merezhkovsky. So he's talking about symbiogenesis as the origin of organisms. By the origin of organisms, he means the origin of species. So how do you get new species? By the combination or the association of two or several beings which enter into these symbioses. And he saw that the major realms of life could actually be distinguished by their levels of symbioses. And that he makes that very clear in this nice paper that Bill and Klaus Kowalik have annotated and translated. But I think so Merezhkovsky himself isn't making really explicit statements about Darwin, but Ivan Wallin is. So Wallin's the person who thought he could culture mitochondria outside of the host cells. Nobody believed him, even in his time, and of course not subsequently. So he thought, well, Darwin got it partly right, that Darwin has found one of the three principles of evolution, but only one of them, and this is natural selection. And what Wallin thought is that natural selection was sufficient to explain the retention or destruction of formed species and only the retention and destruction. He thought natural selection couldn't explain how new species were produced and that something else had to be introduced theoretically to fill this gap. He thought Darwin was looking for something to fill that gap, but that Darwin hadn't found it. So Wallin believed that he and other thinkers along those lines had found this important theoretical principle to fill a gap about the origin of species, and he called it symbionticism. So that's slightly different terminology, but he means the same thing as Merezhkovsky. And he rests the basis of his case for this on mitochondria. So he actually thought there were three principles. He never found out what the third one was, but the third one would guarantee the directionality of evolution to more complex states. And this new edition of his book nicely captures that sort of directionality he was in search of. So there are lots and lots of historical thinkers we could talk about, but what I want to do is bring it into a more contemporary time because there's been a bit of a legacy. And I think the more contemporary people talking about symbiosis as having, some kind of theoretical threat for neo-Darwinian thinking, raise this in terms of three main theoretical tensions. And of course, the person who does this is Lynn Margulis, whom we've heard about several times. I got some of sort of a range of younger pictures here. Here she is in the first one getting married to Carl Sagan. in 1957. The others are all a little bit later. So she says, and she states this all quite clearly, I think not very effectively, but in ways it's worth addressing. So she says the first tension is in terms of population genetics. That symbiosis research shows very clearly that the way in which population geneticists are doing evolutionary biology with these equations is wrong, fundamentally, because the individuals that the equations are supposedly handling are not individuals. So here she's taking a narrow view of individuality, that it has to be sort of, a single genome continuum, that there's only this genetic individuality, not any of the other sorts that have been talked about. And she thinks this problem gives rise to a second problem. The fact that the individuals that are meant to be these countable unities of population genetics do not exist. So, she thinks basically there aren't those entities there. It wreaks havoc with cladistics because you've got composite beings instead. So what she thinks here is damaged is the principle of monophily. So whether one's a cladist or not, at least if you're doing phylogeny, you can agree monophily is important. And she thinks, in fact, this invalidates an entire field of study which she saw as phylogenetics. Now, she starts to talk at this point about the pattern side of things, that when you represent evolutionary trees, once you've included these symbiotic movements, you've wrecked your trees. You can't do it. You've got to get these anastomoses into the tree. Now, I think this is a side issue. She wasn't really concerned about patterns in evolution. She was always concerned about process. So people here, such as Ford and Bill and many others, have addressed these pattern issues, but it's Margulis's treatment of the process stuff that I want to follow up with. So what she's saying here is that because you have these horizontal movements, let's forget the pattern side now, and what we need to worry about is what those horizontal movements are doing. She's saying neo-Darwinian evolutionary theory claims that the only sources of novelty can be the incorporation of random mutations. This was the thing she hated the most. That, recombination, gene duplication, point mutations, all these DNA rearrangements were, she thought, insufficient. And to explain evolution, why? Because they were Lamarckian. So what she's saying there is that anything that moves horizontally has to be understood as Lamarckian evolution, and Lamarckian evolution, as we know, has a very tension-laden relationship, at least historically. But, of course, this has nothing to do with the historical account of Lamarckian evolution. I think what she's doing here is a sort of slight fudge, trying to emphasize the process difficulties. For most people, even very strong people with strong interests in lateral gene transfer, once you get a horizontal acquisition, then you can understand standard Weissmannian evolution. You don't have the normal sort of worries about Lamarckianism there, and I don't think that's the issue anyway. Now, here are just a few contemporary echoes. These were people whose papers John encouraged me to read, and I thought, oh, these are great because they kind of amplify and yet at the same time make a little bit clearer what Margulis is objecting to. The first guy is a biologist, Francisco Carapiso, who's an Azolla biologist, and of course that makes him very interested in endosymbioses of various kinds. So he starts to echo these criticisms. Organisms cannot be divided strictly into separate individuals. Therefore, Darwinism has a problem. He takes that up again with organismal interactions play a central role in giving rise to new biological entities. Well, we know in neo-Darwinian theory that organismal interactions also are very important. They're obviously not creating the kinds of new biological entities that he thinks need explaining. And so the final claim he makes is that it's horizontal processes, so in the same way that Margulis claims, produce the novelty that can actually lead to new levels of biological organization. So sort of ordinary processes, I think they're probably happy with non-horizontal processes. But as soon as you're getting new levels of biological organization, they say you will have to theorize these horizontal movements. Bringing back Jan Sapp again, actually he's not the first author on this paper, but just to make it simple. So he, again, echoes Margulis, so he was quite devoted to expressing Margulis's words, and I think in this paper he makes it very, very clear. the first thing he says is, and his co-authors, there's no individual organism, so therefore Darwinism falls flat on its face because there's no individual selection. Now, whether I don't think any of us will accept that we'll think, oh, the definition here of individual organism is meant to be an excluding one just to make that a true statement. What he says in the end, though, is the point that I'm going to pursue because it connects up with Carapiso's point and also with what Margulis was saying and Wallen and people before. That symbiosis produces variation that's necessary for micro and macro evolution. Now, I think the issue here is actually turning on whether there is an explanatory issue about macroevolution. I'm going to follow up on that, but first I just want to look at some standard counter-objections from contemporary neo-Darwinian thinkers and how they have countered these claims about symbiogenesis. So the first one, which I won't read out, this is Richard Dawkins at the end, partway through a 4-hour discussion with Lynn Margulis. So it's a transcript, that's why his prose is not as precise as normal. So what he's saying is we've got allopatric speciation. That's what all of that says. What's wrong with that? That's sufficient, you know, it's highly plausible, it's economical, it's parsimonious. What more of an explanation could anybody possibly require? Now, you know, apart from sort of thinking there's a lot of selection in that and not much of the other processes going on, the main argument he's making here, and this is still a quote, why on earth would you want to drag in symbiogenesis, he says to Lynn Margulis, when it's so unparsimonious and uneconomical? And I think, unfortunately, this is not actually the right kind of countering example. Argument, because what Margulis can do just completely simply is say, because it's there, because there seems to be an evolutionary impact, because there still seems to be something to explain. That's precisely what she said, and that's why I don't think we can follow the Dawkins line of refutation. Instead, maybe it's more productive to follow John Maynard Smith. Now, Maynard Smith, this quote comes from a book where he contributed a chapter to an edition Margulis put together, so he was feeling friendly enough to be part of that effort. But his strategy is incorporation, you know, just take the sting out of the tail and get on with it. And what he says here is the relevance of symbiosis is that it affords a mechanism whereby genetic material from very distantly related organisms can be brought together in a single descendant, and then you just put that bang into the theoretical machinery, crank the handle, and you've got a perfectly good Darwinian account. Now, I think there's some sort of residue there with this word mechanism, and I'll come back to it. I just wanted to show very briefly that, of course, you can get very, very nice population genetics of endosymbionts, both individually, where you look at, say, Wolbachia or Buchnera or the organelles, and you come up with evolutionary explanations. So once they're there in the cell, once they're actually established in these very stable, reproduced relationships, then you can have very, very adequate neo-Darwinian explanations of what's going on, about whether the evolution of those genomes is being shaped by selection or drift, mutational pressure, what sorts of combinations, why you get different patterns of genome reduction, for example. So we heard some of this already. And you do the same with organelles, you know, working out, for example, whether they evolve non-adaptively. You know, the expectations were that it would be, that it would be non-adaptive evolution, and yet there seem to be instances in which you have to have adaptive explanations of organelle genome evolution. And in some instances, such as phylogeography, you do composite phylogenetics, where you do the population genetics, where you do the population genetics of not just the mitochondrion, but the plastid as well, if it's a plant, and of the nucleus. And then you combine those to get a picture of what's been happening over the evolutionary history. So there's population genetics, despite the symbiogeneticists saying one couldn't possibly do it. But to come back to what Maynard Smith was saying about mechanism. So what he's saying is, once you've got the thing in the cell, you treat it with the standard machinery. what gets the thing in the cell? We just treat any sort of symbiosis, a permanent symbiosis of some sort, or an endosymbiosis in particular, as a macromutation. Now, he doesn't mean here the sort of more mystical, heretical idea of macromutation raised in the 1940s by Richard Goldsmith. He simply means large effect changes in genetic variation. Many of these will be deleterious. Some of them can actually, he thought, be single point mutations with large effects. Sometimes they're going to be many combinations of mutations. He thought this is fully compatible with micromutation. And this is how you describe an endosymbiosis, just as a very large mutation that in some rare cases actually gets maintained in the genome and doesn't cause extinction of the lineage. However, there could be an appearance of discontinuity because of rapid effects, but just an appearance. And I think what this does is throw us into the microevolution versus macroevolution, because one of the reasons for people thinking that macroevolution cannot be explained by microevolutionary processes is because there's an appearance of discontinuity. And it's mostly the paleontologists that have been arguing this, but the symbiogeneticists get into this debate for a somewhat different reason that I'll show you in a minute. So, I'm assuming complete familiarity with microevolution and macroevolution. Mostly people think microevolution will at least take you up to the speciation level. If we're talking about macroevolutionary patterns, we're talking about those sort of speciation patterns at larger temporal and geographical scales. And the issue becomes whether the microevolutionary processes will explain the macroevolution. And for many people, the standard line, so this would be, the neo-Darwinian evolutionary line, that you have mutation, gene flow, genetic drift, natural selection, sexual selection, a few other little processes. Add time, and what do you get? Macroevolution. There is no explanatory problem. And this is Brian Charlesworth here, just making that case very clear. There's no need for qualitatively new mechanisms. He's actually not talking about endosymbiosis, but he could be. And, so he's just making the argument here that the sufficiency of standard population genetics theoretical machinery to explain all the history of life on Earth. Now, there are some more sceptical positions about this that don't reject it. They're sort of in the middle a little bit, where they'll say, these are just people I've kind of picked to represent just because the quote's nice. I don't know if their work is any good. But where they're saying microevolutionary studies generally show that micromutation, genetic drift, selection are capable of contributing to macroevolutionary trends. But the question remains for them and a few other people, not the majority of evolutionary biologists, whether or not microevolution is the major determinant of evolutionary diversification. And this is a matter of debate, not least because of the timescales involved. And by that, they're just meaning the obscuration of how you would actually discriminate at any point. Oops. Yep, so I've got that. Now, here's a philosopher sort of trying to sum this up, but from the point of view of the paleontologist. So this is Todd Grantham. He's a philosopher of paleontology. Philosophers of biology generally have been most interested in large animals, and the debates in the paleontology community. Now, paleontologists, as far as I can understand, are the most likely sorts of evolutionary biologists to be macroevolutionists, thinking that you can't get macroevolution out of microevolution. You need something else. And that's because of the fossil record, the gaps in it. They think those gaps are not just evidential gaps, they're real gaps in the history of life, and a special explanatory mechanism is needed. So what Todd Grantham does is sum up this debate in three positions. There's actually only two, but one is a sort of nuanced version. The second is a nuanced version of the first. So the first one is, as we've heard from Brian Charlesworth, Maynard Smith, and probably Dawkins, macroevolution can be reduced explanatorily to microevolution with no remainder. It's all over. It's fine. Then there's a slightly more nuanced position, and I should confess this is the one I have some sympathy with myself, but I'm not sure I'm going to be able to convey it, but I'll just nail my colours to the mast here. So macroevolution is indeed reducible explanatorily to microevolution, but We need to focus on the macro side, look at those patterns in order to put together the complex combinations of microprocesses that explain those patterns. So it's coming at it from a slightly different direction, even though technically it's the same kind of reduction to microevolutionary processes. And then finally, there's this last position. This is not just the paleontologists. This, I think, is where the symbiogeneticists are locating themselves, and this is their objection to standard Darwinian thinking. They think that macroevolution cannot be reduced explanatorily to microevolution. The paleontologists, at least, believe there's species selection. I think probably 90% of the people in the world who think there's species selection are paleontologists, but maybe there's one or two people here who are species selectionists but aren't paleontologists. But I think they're rare in general. And they think that macroevolutionary mechanisms are special and worthy of study in their own right. So let's go back to Margulis. And these sorts of claims, I think, is where Margulis is identifying herself as one of these she's thinking of irreducible macroevolution. She talks about symbiogenesis, interspecific fusions. She includes gene transfers, especially large ones, and epigenesis. I don't know what she means there. She thinks they're more important than the slow, gradual accumulation of mutations or sexual mergers by recombination. And here, what she's saying is there's that idea of a gradual passage from an ancestral to a descendant species by accumulation of random mutations, so that's the enemy, has not been demonstrated in the field, nor in the laboratory, nor in the fossil record. Instead, what's going to fill the gap? Symbiogenesis. Now, I think that's a bit dodgy, first of all, for the creationist uptake, which has been, no doubt, I think, quite huge, but also because you've got serious problems for symbiogenesis, demonstrating this in a laboratory, even in the fossil record. One can do it by inference, but can't necessarily do it in the ways that she is claiming there. But Margulis isn't on her own. There are other people, here's Brooker and Bordenstein, who use Wallen as their theoretical model. And they do have a more moderate view, I think, because they're willing to agree. In some cases, it can be microevolutionary processes. Other times, you're going to have to think about symbionts. And they think that one can actually understand speciation by symbiosis, not just in terms of very close symbionts, but also in terms of things like the gut microbiota. That if you track those, you'll be tracking the speciation patterns of the animals that contain them, such as these flies here. So what I thought might be a useful exercise is to go to this, in some ways too complex a case, but in other ways the explanation we all would want to have. That if we want to explain the emergence of major biological groups with new capacities, can something like population genetics carry the whole explanatory load? Now, and so what we want is, that transition from Fecker to Lecker, and then maybe even the rapid radiation afterwards. Are we going to be able to explain that by population genetics? So, there are very nice, helpful, illuminating genetic analyses that give a description of the necessary genetic dependencies that show by inference what must have happened in the past to lead to certain groupings of genes and combinations of genes to produce this first eukaryote. But the question is whether it's going to capture all of the causal forces that were necessary to bring about something like that first eukaryote. And it's getting at those causal forces that I think is the issue here, and that I think is ultimately the issue for symbiogenesis. So what's the explanatory focus? Even if we think, well, maybe to get this first eukaryote cell, it's not going to be enough just to track gene frequencies. We need something more in the explanation. Is it symbiogenesis itself that's going to be doing the necessary explanatory work? And it seems to me that in many ways you can make the case that symbiogenesis is a bit more like a condition for some core causal processes driving that evolutionary transformation. And that, so in that respect, symbiogenesis isn't the explanation itself, it's a condition for those explanatory processes. So what would those processes be? And here I think Bill and Nick Lane and John Allen in another situation, have hit the explanatory nail on the head. Because what they're talking here about the transition to eukaryotes in this very well-known paper as hinging on a bioenergetic jump. Now, it's afforded by spatially combinatorial relationships between two cells and two genomes. So that's endosymbiosis. But that's not the explanation. The explanation is a bioenergetic, a metabolic explanation, rather than purely a natural selection acting on mutations accumulated gradually. So the way that I would frame that is that you're making a shift in that kind of explanation from a population genetics explanation, which is gene-centric, to another kind of explanation that works in concert with it. Maybe in some cases outweighs it, other cases might not outweigh it. And that eukaryogenesis and other major endosymbioses require metabolism-centric explanations. That there has to be some different explanatory balance at particular periods in evolutionary history. It doesn't invalidate the gene-centric explanations, but to get the right causal processes, there's going to have to be something more. Here's Harold Morowitz talking about this explanatory balance, that there's more to metabolism than can be captured in the selfishness of genes or compartments, and that we may, in fact, in many instances, want to think of metabolism as more than a follower of genes. In many respects, we'll want to think of it as a leader. Winding up, yep. So, and here's Victor Di Lorenzo, who I think puts in a general way exactly what John Allen was saying yesterday, that there are instances where we actually need to understand the metabolism as having the explanatory force in particular situations, that you have biochemical networks with their own autonomous drives which constrain the execution of instructions from DNA. So one still maintains this DNA makes RNA makes proteins, there's no problem there, but it's in a feedback loop where in some instances you've got metabolism not just constraining but feeding back into that DNA makes RNA makes protein process exactly what can happen. And I think that redox control instance yesterday is a very, very nice example of that. So we get a change of explanatory focus if we contrast standard, let's say population genetic or neo-Darwinian theoretical accounts with what we might call endosymbiotic, but the focus in the endosymbiotic explanations is on metabolism. So we move from a gene-centric explanation to a metabolism-centric one, from an informational one to one about biochemical networks, from populations of organisms with very similar genetic complements to communities with often very different ones, and yet those relationships are maintained and instructed by the metabolic networks. So there are other potential examples. So I use the eukaryote one because it was put so nicely in Nick and Bill's paper. There are other ones. I think you can explain some mass extinctions metabolically better than you can with gene by tracing gene frequencies. Not all mass extinctions, only some, but the Permian's a major one. You know, we can look at this paper that also was done by Nick Lane, the divergence of bacteria and archaea. One can come up with a metabolic explanation for that. And I think something like the origins of life is a paradigm case of the complementarity of these two explanatory strategies, where, you know, it doesn't matter what comes first or last. It doesn't matter if, in the case of eukaryotes, whether mitochondria are first or last. At some point in that process, you've got to combine gene-centric and metabolism-centric explanations. And as Morowitz continues, now I don't think this is the case, but I think it's worth mentioning, he thinks that there may be a grand unified theory for at least the core of metabolism, and it links ecology, biochemistry, and the Darwinian world into a whole larger than any one of them. But whether or not that's the case, and there aren't many signs of it at the moment, there are particular explanatory instances where one needs a metabolic explanation to accompany the neo-Darwinian one. And that is what I think the sort of symbiogenesis objections are pointing us towards. Is there feedback into microevolution? Yes, there is, in cases where you get these metabolic feedbacks into actually what the DNA can do. Do we have a fully fledged alternative theory? No, we don't, but we've got a supplementary explanation. Is it symbiogenesis? I don't think so. I think it's more likely to be some sort of metabolically informed supplement to to microevolution, but it's not, in many instances, going to be an optional extra. And at the very least, things like major transitions need adjustment to include more than informational and individuality outcomes. Just to conclude, this bottom part, I thought I'd end up with an endosymbiotic theory of philosophy and science. We've heard a lot of bits of philosophy popping up in the science talks. And I think we can imagine that, once upon a time, way back in the early history of modern science, there was some kind of greater equilibrium between philosophy and empirical science. Now, at some point, maybe in the last century, or the century before, we had philosophy kind of engulfed by the science. But the one thing that seems to have happened is that no matter how you do science, how long you keep doing science, doesn't look like you're going to get rid of the philosophical issues. That they're always going to have some kind of feedback into the science in this kind of way. And I think this is what this very nice topic has at least demonstrated. So these are some acknowledgements. I want to very much thank John and Ford for talking through some of this. Okay.